Hi there. Today we'll be talking about skeletal muscle contraction, and we'll be taking a quick look at the anatomy of the skeletal muscle. Skeletal muscle is simply a muscle that is attached to the skeleton, and uh, it's attached to the skeleton by means of the tendon. The muscle is surrounded by a connective tissue sheet known as the epimysium, which you can see surrounding the muscle, and the muscle is divided into fascicles. These are fascicles. So the muscle is divided into fascicles by means of another connective tissue called the perimysium. All right. Now, each fascicle, as you can see here, is composed of individual many muscle fibers, which are muscle cells. So we refer to muscle cells as fibers. These muscle cells are multinucleated. They have many. They have more than one nuclei, which you can see here. This is one nucleus. This is another. This is another. This is another, and so on. So the um, the muscle fibers or muscle cells, they are also separated by another connective tissue called the endomysium. Muscles are referred to as effectors as they cause an effect to happen. The effect is usually movement, and muscles are every muscle fiber is innervated. It has a connection to a neuron, and these neurons that innervate muscles, they are referred to as motor neuron. And the point where the motor neuron and the muscle fiber meet is referred to as the neuromuscular junction. Okay, this illustration shows the neuromuscular junction. This is the axon terminal of a neuron, uh, which is in close opposition to a muscle fiber. There are physiological interactions between the neurons and the muscle fiber. Now, the a single motor neuron and all the muscle fibers that it stimulates is referred to as the motor unit, which is the functional unit of the skeletal muscle. Now, this shows an illustration of a motor unit. In this case, you can see a single neuron innervating seven muscle fibers. All right, so this is a motor unit. Okay. Now, individual muscle fibers are referred to as myofibers. They are multinucleated, as we've said earlier, and the multinucleation is caused by the fusion of immature myoblasts during the development of the muscle. Immature muscle cells called myoblasts, several of them fuse together to become one cell, right? While still retaining the individual nuclei from all of these cells, and so you have one cell that has multiple nuclei, and the each individual muscle fiber or muscle cell contains hundreds to thousands of myofibrils arranged in parallel. You can refer uh, to the illustration of the anatomy of the muscle earlier. Each fiber is innervated and the fibers are organized into motor units. The myofibril um, contains actin filaments. Remember that we said that each muscle fiber or each muscle cell is also composed of hundreds of myofibrils. All right, and the myofibrils in turn is made up of filaments, the thin filament or actin filament, and the thick filament or myosin filament, which are partially interdigitated. It means interdigitated because these filaments they run they okay they have a relationship, they run in amongst themselves as if we were uh, putting the fingers of both hands into amongst themselves. We'll see examples. We'll see an illustration of what we're talking about in a slide from now. Now, each myofibril is made up of 3,000 actin filaments and about 1,500 myosin filaments, and each myofibril is composed of sarcomeres, which are connected end to end. Now, let's take a look at the sarcomere. Now, the sarcomere is the contractile unit of the muscle. Um, it is the smallest functional unit of the skeletal muscle fiber, um, and it consists of a highly organized arrangement of several types of proteins. We have contractile proteins, regulatory proteins, and structural proteins. Um, there are there are three bands in this sarcomere. Okay, now earlier I talked about how the actin and myosin bands, the thin and thin and thick filaments, how they interdigitate. Now you can see what we're talking about here. This the filaments illustrated in red, they are the myosin filaments. You can see how they run, how this myosin filament runs in between two adjacent actin filaments, and in turn each actin filament also runs in between two adjacent myosin filaments. So that's what we mean by interdigitation. Okay, so there are three bands in the sarcomere. We have what we refer to as the I band. The I band consists of only actin. So you can see the I band. Okay, what we are looking at is a single sarcomere. All right, so we can see the I band that it consists of only actin filaments. Okay. Then there is what we refer to as the H zone. The H zone only consists of only myosin filaments. Then we have the A band. So this is the H zone, which you can see here, and we can see clearly that it consists only of myosin filaments. Then finally we have the A band. The A band, which extends from here to here, which composes of both actin and myosin filaments. So a single sarcomere contains one dark A band with half of the lighter I band on each end. Again, let's go back to the previous slide. So we said a single sarcomere contains of one dark A band. Yes, this is the A band. Okay, with half of a lighter I band on each end. So this is half of an I band. This is another half of an I band on each end. All right. So this from this area to this area, this is the sarcomere, right? You can see the A band. You can see half of the I band here, half of the I band there. You can see the H zone, which the H zone, like I said, consists of only myosin. The I band consists of only actin, whereas the A band consists of both actin and myosin. Okay. So the striated appearance of skeletal muscle is due to the arrangement of this actin and myosin filament. Okay. So we've already said this, that the A band consists of only myosin, the I band consists of only actin. Okay. So now let's continue looking at the structure and organization of the skeletal muscle. All right. So again, a single myofibril, which is, as we've said, composes of what? Actin and myosin filaments. All right. And we said that a single muscle fiber consists of hundreds of myofibrils. So we can see how this illustration has broken down the relationship of a myofibril to the whole muscle itself. Hundreds of myofibrils make up a muscle fiber. Hundreds of muscle fibers make up a fascicle, and so on. All right. So when we now look at the myofibril under the microscope, yes, this is what we might see, and it's still the same thing as what we saw in this illustration here. Okay. So again, we can see the I band, right? I band, which is made up of only actin filaments. You can see the H zone, which is made up of only myosin, and we can see the A band, which is made up of both actin and myosin. Okay, so we've already said this. We're going to skip. We'll skip this, as we've already said this several times. Now, the hundreds to thousands of myofibrils can be found inside one muscle fiber. Again, what we've said earlier, and each myofibril consists of thousands of sarcomeres. All right. So as we've already defined what the sarcomere is, we said that that a single sarcomere contains one dark A band with half of the lighter I band on each end. So let's come back here. Right, a single sarcomere contains one dark A band, right? A single A band with half of the lighter I bands on each end. So this is half of an I band. This is another half of an I band. All right. So along with the A band, the A band extends from here to here. Then this is the I band on this side. Then another I band on this side. So that composes the sarcomere. Okay. All right. So it is the shortening of these individual sarcomeres that leads to contraction of individual skeletal muscle fibers. And during the contraction, the myofilaments slide along each other so that the distance between the Z discs shortens and resulting in the shortening of the sarcomere. Um, we'll talk more about this later. Now, the length of the A band does not shorten during contraction, but the H zone and I band regions shrink. So let's go back to have a look at this. Okay, so we said that the okay, let's look at the okay. So during contraction, so what happens is that these filaments, these actin and myosin filaments, they move relative to each other, such that you can imagine if this actin filaments, which are illustrated blue here, if they were to similarly approach each other, it will result in a situation where uh, the H zone, right? The H zone will shorten, but the H zone and the I band will shorten, but the length of the A band will not it will not shorten, because these filaments will slide along each other during contraction. So, but we'll see what we're talking about later. All right. So the length of the A band does not change because the thick myosin filament, it remains a constant length, but the H zone and the I band regions will shrink. All right, and these regions, they represent areas where the filaments do not overlap, and as the filament overlap and as filament overlap increases during contractions, these regions of no overlap will decrease in size. So let's have a look at this. So we are saying that at rest, when the muscle is not contracting, if you come to the high band here, you can see in the high band, that the actin and myosin filaments do not overlap in the high band on both sides. But in the H zone, you can see the H zone illustrated here. In the H zone, actin and myosin filaments also do not overlap. All right. So, but during contraction, during muscle contraction, there will be an increase in overlap of the two filaments, actin and myosin, such that those areas of the sarcomere where there was no overlap of the two filaments, there will now be overlap because of the way the two filaments will move relative to each other, and so it will lead to shortening of the I band and H zone. However, the length of the A band will not change. Okay, so these are still illustrations showing what we've talked about, the the sarcomere, the various bands and the zones. Okay. So this also this shows and we are still looking at the actin and myosin filaments of the sarcomere more closely. So now in this particular illustration, we can see that the myosin filaments are colored purple here. So this is an example of the myosin filament. And if we take a closer look at the myosin filament, we can see that there are some projections which are called heads on the myosin filament. Remember that the myosin filament is also referred to as the thick filament. All right. So these heads, we can see a closer look of the head. Right. The head has a hinge. A hinge is that thing that makes a door to that attaches a door to the door frame, and along which the door turns in or out. All right. So there is a hinge. There's a region here referred to as the flexible hinge region, which allows this head to move, to move in this direction, then to move in this direction, then move back. As if it's a door moving back and forth along its hinge. All right. So this is a myosin head. Okay. All right. So now let's talk about the sarcoplasmic reticulum. The sarcoplasmic reticulum is simply the endoplasmic reticulum, right, of skeletal of muscle cells. The endoplasmic reticulum of muscle cells is referred to as sarcoplasmic reticulum. All right. So it's a specialized type of smooth endoplasmic reticulum in muscle cells, and it surrounds the myofibrils. Right. The sarcoplasmic reticulum stores and releases calcium ions, because calcium is necessary for muscle contraction, and so the calcium is stored within the sarcoplasmic reticulum inside the muscle cells, and is released when needed. Okay, so if you look at the illustration here, this is the sarcolemma. That's the cell membrane of muscle cells. It's referred to as sarcolemma. All right, and we can see this sarcoplasmic reticulum, which is illustrated and labeled here. Okay. Now we can also see what is called terminal cisternae, which are large sacs. These are the terminal cisternae. They are part of the sarcoplasmic reticulum, and it's inside them that the calcium is actually stored. Now there's something referred to as the transverse tubule system, or T-tubule system. Okay. So these are just invaginations of the sarcolemma. So okay, let's come back here now. This is a plasma membrane of the skeletal muscle cell, right? So you can see holes where the sarcolemma invaginates, and it forms a kind of system of tubes. This hole that invaginates from the plasma from the sarcolemma now forms a system of tubes that penetrate deep into the muscle. Okay, so that is the transverse tubule, and so you can see that the transverse, because this tubule, it's like a pipe, a tubule, right, that runs deep into the muscle, and it's connected to the outside of the cell, because as you can see here, that the tube is connected to the outside of the cell here. All right. Okay. So the transverse tubule ensures that the action potential on the sarcolemma can get to the interior of the cell and close to the sarcoplasmic reticulum throughout the sarcoplasm. Again, sarcoplasm is just the cytoplasm of the skeletal muscle cell. It's referred to as the sarcoplasm. Okay, so this illustrates what we're talking about. So if you have action potential on the sarcolemma, it can penetrate deep into the interior of the muscle cell by moving along the transverse tubular system. Okay, and of course the transverse tubular system, it runs along it runs along the terminal cisternae. You can see how the terminal cisternae is closely opposed to the transverse tubule. So it means that once the action potential is running down the terminal cisternae, the action potential can easily get to spread to the terminal cisternae, and eventually cause the terminal cisternae to release the calcium when the calcium is needed. Okay. All right. So now we'll talk about the types of muscle protein. We have contractile proteins, and we have regulatory proteins. Of the contractile proteins, we have two. actin, which is referred to as the thin filament, and myosin, the thick filament. Now, for the regulatory protein, we have troponin and tropomyosin. Troponin is excitatory to contraction, whereas tropomyosin inhibits contraction. So now this illustration shows what we are talking about. You can see the way um, the for the actin filament, right? This so we can see that what we call the actin filament uh, or the thin filament is not does not compose of only the protein actin. There are some other proteins, right? We can see the tropomyosin. Uh, can see the tropomyosin illustrated there. You can also see troponin. All right, and whereas you can see the this is the myosin filament with the heads, which we've already discussed about, which we've already discussed earlier. So you can also see another actin filament. You can see so this particular do we say dark brown molecules are actin, right? Whereas these are troponin. These here are troponin. These are actin, and these are tropomyosin as illustrated above there. Okay. So now, let's talk about the proteins of the thin filament. Yes, we have the actin, which are composed mainly of G-actin molecules, and as well as G-actin molecules which attach to each other to form F-actin strands or chains. All right. So, in illustration of what we are talking about is here. We can see actin molecules which attach to each other to form actin strands or chains. All right. And the actin chains, they twist around each other like the twisted double strand of pearls. Right. So we can see the way they twist around each other, and that was what was illustrated here too. They twist and twist and twist around each other. Okay, so we also have tropomyosin, right? And two strands of this protein twist around actin, which you can see here. Two strands of tropomyosin. One tropomyosin is the dark brown thin strand here. So two strands of it twisting around the two strands of actin. So you can see two strands of actin, one two twisting around each other, then twisting around these actin strands. We now have two strands of tropomyosin. Okay, and the tropomyosin covers the sites on actin where myosin will bind. Now, so during muscle contraction, at some point, the myosin heads, which we talked about earlier, will bind to some specific sites on actin. All right. So, but until the contraction is about to happen, those myosin binding sites, those sites on myosin those sites on actin to which myosin will bind during contraction, they are usually covered by tropomyosin, so that they are not available for binding by myosin. All right. So finally, we have troponin, and this protein in its turn is attached to both tropomyosin and actin, and is composed of three. So okay, let's look at the example of the troponin. So in this, looking at this is a troponin molecule, and we can see how it is attached. Right, it's attached to both actin and tropomyosin, and the troponin is composed of three distinct polypeptide complexes: troponin I, troponin T, and troponin C. All right. So troponin I binds to actin, troponin T binds to tropomyosin, and troponin C binds calcium to initiate muscle contraction. So C for calcium, T for tropomyosin, and troponin I binds to actin. Okay. So this shows what we're talking about again. You can see the troponin complex, right? It's a complex of three different polypeptides, right? Troponin I, troponin T, and troponin C. Okay. So this is a summary of what we've been saying in the last few slides. We can see how uh, um, G actin molecules are attached together to form chains, which are the F actin strand or chain, and two actin strands wind around each other. They spiral around each other, right? Then there are also two tropomyosin strands, 1 and 2, also winding around these two actin strands. Then we can see troponin complexes which are attached to both actin. You can see these troponins attached to the actin strands and also attached to the tropomyosin strand. Okay, so uh, G each G actin molecule has a binding site for the myosin head called actin active site. So let's go back here. Now these dark spots on each actin molecule, they are what we refer to as the actin active sites, and it is to this active sites that the myosin heads will bind to later during the course of contraction. So, but we can see that when contraction is not happening, that uh, tropomyosin, which is this dark brown strand, covers those actin binding sites. So when contraction is not happening, the myosin the myosin binding sites are covered. The myosin binding sites on the actin molecules are covered um, by tropomyosin. Okay. All right. So these active sites are covered and hidden from the myosin head by the inhibitory protein tropomyosin. That's why we said that tropomyosin is inhibitory, because it prevents myosin from binding to actin, which is a necessary step for muscle contraction, and so it is inhibiting contraction. So, and under resting conditions, no calcium is bound to troponin, and that will be troponin C. Troponin C is the part of the troponin complex that binds to calcium. All right. So under resting conditions, no calcium is bound, and this prevents actin and myosin from interacting. When calcium binds to troponin C, then the tropomyosin complex moves, thereby exposing the actin binding sites for myosin. So, so this just tells us that before contraction can happen, right, the calcium is essential. It must bind to troponin C, and this will cause the tropomyosin. Remember that troponin as a whole is connected, is bound to both actin and tropomyosin, right? And so when calcium binds to troponin C, it causes the tropomyosin complex to move, thereby exposing the sites on actin for which myosin will bind, to which myosin will bind. All right. So now let's talk about the proteins of the thick filament. Yes, we've already talked about it earlier. So, but the head also which has a hinge or the joint and a tail as well too. Okay, so this is how it looks like: the head, hinge, and the tail. Now, each myosin head contains a an ATP binding site, adenosine triphosphate binding site, as well as an ATPase enzyme, adenosine triphosphatase enzyme. Because we know that muscle contraction is an energy muscle contraction requires energy, and this energy is supplied in the form of adenosine triphosphate, and so there's also an enzyme, that's the ATPase, adenosine triphosphatase, on the myosin head, which will break down the ATP molecules to generate the necessary energy. Okay. So, this is illustrated here. that, okay, now this is the head of the this is the myosin head, and this is part of the myosin head that will bind to the actin. All right, then this is the ATP site, where ATP will be broken down. All right, so this is shown more here: ATP binding sites, actin binding sites, and so on. All right, so ATPase, adenosine triphosphatase, is the enzyme which degrades ATP into ADP plus phosphate with the generation of energy. All right, and it's that energy that powers muscular contraction. Now, um, characteristics of the thick filament. Myosin has ATPase activity, which you've said, and when the ATP is split, when the ATP is degraded by the ATPase, the energy released puts myosin in a high energy state and also increases the affinity of myosin for actin. Okay, all right. So, when the myosin binding site on actin has been exposed, remember that we said that exposure will only happen when calcium binds to troponin C, right? And uh, the so it means that muscle contraction requires two things. It requires calcium and it requires ATP. The calcium will bind to troponin C and cause exposure of the site on actin to which myosin will bind. That's one criterion. Then the second is that ATP will bind to myosin, and it will then be split by ATPase, all right, to release energy, and this energy will put myosin in a high energy state and also increase the affinity of myosin for actin. So subsequently, the myosin heads will now the bind to actin, which, remember what we said calcium will lead to. All right, it will lead to exposure of the sites on actin to which myosin head wants to will bind to. All right, so once this happens, the chemical energy is transferred to mechanical energy, and the myosin pulls the actin filament. This generates active tension in the muscle, and this is referred to as the power stroke. Now, if the force generated by the power stroke is sufficient to move the load, then the muscle shortens. This is referred to as isotonic contraction. If the force generated is not sufficient to move the load, then the muscle does not shorten, isometric contraction. Now, when there is a theory that explains that how the interaction of actin and myosin, how the interaction of these two contractile proteins leads to contraction. Okay, so what happens? So it is called the sliding filament or the walk along theory. Okay, and so what happens is that when for contraction to occur, actin and myosin filaments, they slide along each other such that the distance between the two Z disc will decrease. Distance between the two Z disc will decrease. The high band gets smaller, but the A band does not become smaller or bigger, as we've said earlier. All right, so we can see what we're talking about here. All right, that the now look at the high band here. When these filaments slide along themselves, you can see. Okay, look at the ends of the actin filaments on both sides there. When the muscle is relaxed, but when they are pretty far apart, the H zone is at its normal length. But when the muscle contracts, the two ends of the actin filament will come closer to each other, and the H zone shortens. The length of the H zone shortens, as we can see when the muscle is contracted. All right. So the same thing happens here. When that happens, the high band too, when the muscle is relaxed, the high band has a is thicker than when the muscle is contracted. Okay, so during contraction, what happens is that the heads, the heads on the myosin filaments, they attach to actin filaments. Then the power stroke causes the the heads to drag the actin filament backward, to drag the actin filament in this direction. Then eventually, the heads will detach, go back to the resting state, and bind to the actin filament again. Then drag the actin filament again. Okay, so this will lead uh, to contraction. Okay, now an animation that will illustrate this. Now look at this. So first, the myosin heads bind to the actin. Then, okay, so the myosin head binds to actin. Then they drag the actin. Then it will release it. Then bind to it again and drag it again. All right, and so this is what causes the shortening of muscles that we refer to as a contraction. The movement of the actin and myosin filaments is caused by myosin heads dragging the actin filament in one direction. Okay, so this is what happens. So the animation that I just showed is what is explained here. So myosin first binds to actin molecules, then it pulls the actin filaments, then it detaches and returns to its initial state, then binds to the actin filament again, and also pulls it again for a second time, and so on and so forth. So attachment of myosin to actin activates the enzyme ATPase in the myosin head. The ATPase breaks down ATP and releases energy, and the energy is used in a power stroke to move the myosin head. All right, so we have a situation where at the start, before contraction, we have some okay, the situation we have is that first of all, calcium has not bound to troponin C, and because of that, the sites on each actin molecule that the myosin heads will bind to, those sites, they are covered by tropomyosin, and so uh, the myosin heads cannot bind to actin, and so contraction cannot occur. All right, so the first thing for contraction to happen is that calcium will bind to troponin C, right, and that will cause some changes that will result in the tropomyosin that was blocking the binding sites on actin, to which myosin will bind to. Those troponin C will move out of the way, and this will allow myosin to bind to actin. All right, so that's the first thing. The first step is that calcium binds to troponin C. Then tropomyosin moves to expose the myosin binding site on actin. Then myosin then binds to actin. Okay. Then the next step is that the ATP will have bound to myosin 2. Then ATPase on myosin will break down the ATP to produce energy. It's that energy that the actin heads will use to drag, what we refer to as the power stroke, to pull and drag the actin and cause the sliding of actin on myosin. All right. So this is a summary of the sliding filament theory of contraction. We've said all of this, but can just have a look at it. All right, so let's now talk about the general mechanism of skeletal muscle contraction. So it involves two things. First is the neuromuscular transmission. Before a muscle can contract, it must be stimulated by it must be stimulated by the neurons, by the motor neurons. Then after that stimulation is what the next thing is now the excitation contraction coupling, which we've talked about more or less in the last few slides. So let's first talk about the neuromuscular transmission. Alright, so the process of muscle contraction begins at the site where a motor neuron's terminal meets the muscle fiber. That's the neuromuscular junction. Okay, and we've said that every fiber is innervated by a motor neuron. Okay, and excitation signals from the motor neurons, we activate the skeletal muscle to contract. Okay, so now let's talk about the process of neuromuscular transmission. An action potential moving along a motor neuron. When it reaches the end of the motor neuron, it will when it reaches the end of the motor neuron, uh, what does it do? It will okay, let's come here. Now the action potential reaches the end of the motor neuron. Okay, all right, and the uh, so okay, first of all, this is a neuron and this is a skeletal muscle. All right, so once the action potential reaches the end of the motor neuron, it stimulates calcium channels in the motor neuron to open, and calcium enters into the motor neuron. Okay, now the influx of calcium into the motor neuron will cause vesicles that contain the neurotransmitter acetylcholine. Those vesicles will move to the membrane of the to the synaptic uh, to the membrane of the neuron. The vesicles will fuse with the membrane of the neuron and release acetylcholine into the synaptic cleft. Okay, the acetylcholine diffuses across the cleft to the uh, skeletal muscle, right? And once they get to the skeletal muscle, the acetylcholine will bind to their receptors on the membrane of the muscle fiber. So acetylcholine binds to the receptor. Now those receptors, those acetylcholine receptors, they are also they are attached to sodium ion channels. So once the acetylcholine binds to their receptor on the sodium ion channels, it will cause the sodium ion channels to open, and sodium enters into the muscle cell. Okay, so the sodium will thus depolarize the membrane of the muscle fiber, and this will cause a graded potential called an end plate potential. So potential this end plate potential will pass into the T tubules. So if you look at picture number 4 here, all right, the potential that is generated here after sodium, we said sodium will enter into the the muscle cell and depolarize it. Now that potential will move along the membrane of the muscle fiber, initially along the membrane, then it will penetrate deep into the muscle fiber, into the muscle cell, along the T tubule system, the transverse tubule system. Okay, so once it passes through the T tubule system, it will eventually the potential will reach the endoplasmic reticulum, right? The terminal cisternae of the endoplasmic reticulum, and it will trigger the endoplasmic reticulum to release the large quantities of calcium stored within it, right? Because of the activities of voltage gated calcium channels. All right. So once this calcium is released, remember that we said that for muscle to contract, that the first step is that calcium uh, must bind to troponin C, right? So that calcium that binds to troponin C, this is where the calcium comes from, and it's a direct uh, result of the action potential that comes down along the motor neuron, causing the release of acetylcholine. Acetylcholine eventually causes the depolarization uh, of the membrane of the muscle fiber, and eventually leads to release of calcium from the terminal cisternae and sarcoplasmic reticulum. Okay. All right. So after that, next, so those what we just described was the events at the neuromuscular junction, right? Neuromuscular transmission. So the next step is to talk about excitation contraction coupling, which we've already been talking about earlier, but let's just repeat it. All right. So the excitation contraction coupling is the physiological process of converting an electrical stimulus, that is an action potential or a graded potential, to a mechanical response, either an action potential or more specifically the end plate potential, to a mechanical response, um, which is a muscle contraction. Okay. Okay, sorry. Let me say that again. End plate potential is excitation contraction coupling is the physiological process of converting an electrical stimulus, that is the action potential that comes from the motor neuron, to a mechanical response, which is the muscle contraction. Okay, so and what happens, we've said some of it earlier, is that the end plate potentials that is generated in the muscle cell membrane will depolarize the T tubules. The T tubules will depolarization of the T tubules causes the calcium channels in the nearby sarcoplasmic reticulum to open, all right, and release of calcium into the intracellular fluid. And when intracellular calcium increases, the calcium binds to troponin C on the thin filament, causes a change in troponin that moves tropomyosin out of the way of the myosin binding sites on actin. All right, and thus myosin can bind to actin, and the cross bridge cycle begins. Okay. So at first, no ATP is bound to myosin. The myosin is tightly attached to actin. Then ATP then binds to myosin, produces a change in myosin that causes myosin to be released from actin. The myosin head is displaced towards the plus end of actin. The ATP is hydrolyzed to ADP and pi. Then the myosin head attaches to a new site on actin, forming a cross bridge. ADP is then released. The myosin cross bridge rotates towards the center of the sarcomere, and this constitutes the power generating stroke. That's the power stroke. So let's go back to the animation that we showed earlier. All right, so that's the power stroke right there. Then it repeats itself, a power stroke. So initially, there was no once calcium causes exposure of the actin, indirectly causes exposure of the myosin binding sites on actin. The myosin will bind to actin. Then once the ATP is degraded to yield ADP and energy, that energy will be used by the myosin to cause the power stroke that pulls and drags actin along. Okay, so the myosin head binds ATP again, and the cross bridge detaches from actin. The cycle repeats as long as calcium is bound to troponin C. Each cross bridge cycle thus walks myosin further along the actin filament. And it is the formation of these cross linkages between actin and myosin and the sliding of the thin on thick filaments that produces muscle shortening. So this is what we're talking about again. If we look at this illustration to illustrate the cross bridge cycling. So myosin heads hydrolyze ATP. You can in one here, myosin heads hydrolyzes ATP. They become reoriented and energized. Okay. Then the myosin head binds to actin and forms a cross bridge. Then the myosin cross bridges rotates towards the center. So that rotation is what drags the actin along. Then myosin binds to ATP. The cross bridge detaches from actin, and the process uh, continues over and over again. So this is a summary of the events of excitation, contraction, coupling. And okay, this is a summary. This is another summary of the events. Again, another summary of the excitation, contraction, coupling. So what happens after acetylcholine binds? Action potential. Okay, this is not the potential generated is it's strictly called the end plate potential, right? And travels down the T-tubule, causes release of calcium. Calcium binds to troponin, cross bridges form between actin and myosin, and so on. Okay, so now let's now talk about what happens during relaxation. So usually muscle contraction stops when signaling from the motor neuron ends, right? When the motor neuron is no longer stimulating the muscle fiber, then this leads to repolarization of the, remember that the acetylcholine that bound to the muscle that bound to receptors on the muscle led to influx of sodium that depolarized the muscle. All right. So, but when the motor neuron is no longer is no longer when action potential along when signals from the motor neuron stops, all of this stops, and it will lead to repolarization of the muscle fiber, repolarization of the muscle fiber, and the calcium channels in the sarcoplasmic reticulum will be closed, right? So okay, let's go back to the previous. All right. So the calcium that was released will stop being the release will stop, and it will be reabsorbed back into the sarcoplasmic reticulum. Okay, calcium are pumped back into the sarcoplasmic reticulum by sarcoplasmic reticulum calcium ATPase SACA. Okay, and this and once calcium is no longer present at the is when once calcium is no longer bound to troponin C, then tropomyosin will recover the binding sites on actin. Okay, and the muscle fiber loses its tension and thus relaxes. So we have come to the end of the lecture. Thank you for listening.